everyone, and thank you for joining us today. My name is Chloe, and I'll be your host for this Precisionary Instruments webinar. We're delighted to welcome Ken Shumel, Staff Research Associate 2 at the University of California, Davis. Ken brings over 30 years of experience in mammalian cell culture and has played a central role in advancing precision-cut lung slices, or PCLS, methods for both human and non-human primate tissue. His work in the Zeki Lab focuses on applying PCLS models to assess airway smooth muscle contractility, drug responsiveness, and therapeutic development. Today, Ken will walk us through the fundamentals of preparing and maintaining lung slices and highlight real-world applications of this model in pulmonary research. As a reminder, feel free to submit any questions in the Q&A box during this talk, and we'll address them after the session. And with that, I'll turn it over to Ken.
Everyone, my name is Ken. Last name is Shumel. I'm at UC Davis. I have 32 years of experience in cell culture and several years with slices. I'm here today to present practice and theory of precision cut lung slices for all of you. And we have a slide presentation here. To begin, I would like to talk about the overall idea of precision cut slices. What we have is various methods in medical research through which we can obtain information and samples for analysis to do research. And you can see the six basic models. I don't have, one thing I don't have is my cursor.
It should be at the bottom left of your screen. It should show up. Yeah.
Oh, okay. You see my cursor now?
Yes.
Okay,
You
good.
can also do like a laser pointer if you want to at the bottom of your screen, but we can still see your mouse, so that's fine.
Okay, all right. All right, so everybody knows we can do pure in vitro biochemistry to do scientific research. So compare an activity in a living cell or living organism to biochemistry. We can do cell culture. There's animal experimentation that we can do. There's clinical subjects. So you can obtain samples from the hospital, from a hospital patient, and analyze those. There's computer modeling. A biological process can be modeled on a computer if you know enough about it. And today, another emerging method is ex vivo, which can be organs, tissues, but in this case, we'll be discussing slices. So all these methods are commonly used in laboratories across the country, and the slices have become a very powerful tool. And we'll explain why and how they are used. So to start, we have a precision cut slice here, and this is a lung slice. And the beauty of the slice is in the smooth muscle. So we have a smooth muscle ring. Just to jump back really quick, you can see a slice. This is a slice from a mouse that if you look at the, what you mostly see is alveolar parenchyma using up most of the surface area of that slice. And you'll see various areas of smooth muscle rings. And you'll have occasionally some blood vessels that you might see in the slice. So you have alveolar parenchyma, smooth muscle, and there'll be some epithelium here inside the smooth muscle ring. But that is essentially capitulates the area of the slice and how it looks under microscopy. Inside the alveoli, you may have interstitial tissue between the various alveoli that is connective tissue. And the smooth muscle, the beauty of the slice is not in the alveoli, because those will mostly be shredded during the slicing process because they're so large. The epithelium is here along this interior. It's very, and there's not much epithelium there relative to the mass of the slice, but the smooth muscle rings are substantial. And this is under 10x microscopy. So that's basically composed of slice. Now here we have a picture of a slice, and you'll see how the slice smooth muscle ring is typically associated with a blood vessel that runs alongside of it. And again, you see parenchyma and some air spaces here. But this is typically how the slice will look, and this is what we are interested in is the smooth muscle ring, because that is a functional part of physiology that exists within the slice. And so it is different from cell culture in that in cell culture you have an incomplete organ system. You can only recapitulate the epithelium or one cell type. Occasionally you can do co-culture, which is somewhat impractical because you'll need different types of media for the different cell types, and you'll have a severe loss of total organ histological architecture, which is a problem with cell culture because you only have one type of tissue, one cell type. With animals, you'll have a limited number of data points, because you can only run so many animals at one time. Unlimited experimental flexibility, and you will also have influences of systemic physiology on the experiment. So particularly with the endocrine system, but there's also substantial costs of financial costs and labor involved in managing all the animals. So the slices make this much easier in that you can do good science with many experimental groups, and you don't have to manage all those animals. So that gives us a huge experimental advantage, which is why tissue slices are becoming more and more popular. So here we have a nice smooth muscle ring, and again, you can possibly see an epithelial layer inside. And what I'm going to click, hopefully I can click, you can see if I click this video, what I want you to look at is the cilia. So I'm going to start this video. Do you see all these cilia here along the interior of the slice? And this just gives you, so here's your smooth muscle ring. And the cilia beating is indicative of the viability of the slice itself. It tells you that you have a healthy slice, and it usually will show up after days of culture. So once you've generated your slices, and they are in culture for some days, the cilia will rebuild, and they may be active and beating, which is indicative, as I said, of a very healthy slice. So this is when you do this is in mouse. It will also happen in monkey or human, or it should happen. You should see cilia beating with any animal. But this is a nice example of a healthy slice and what you would like to see when you have good clean slices. Okay, so the regions of a slice have a cross-sectional of gross lung anatomy. You'll have, as I said, lung parenchyma, just to summarize, that will be shredded. If you want to study alveoli, you will use organoids, not slices. So you'll use a different experimental model. The epithelium is intact, but is a small cross-sectional fraction of the slice. So you'll use air for that. It's best to use cell culture, air liquid interface cell culture, and the conducting airways are contractile, and it's a substantive component of the slice mass. So they are very useful in the slice to assess, and there'll be some underlying matrix on to go back under here. You'll have some underlying matrix between the smooth muscle and the lung parenchyma, and some underlying matrix here between the epithelium and the smooth muscle. But it's not very prominent under microscopy. So under microscopy, what is prominent is the alveoli, which are shredded, and the smooth muscle rings, which are physiologically contractile. So if we are going to do slices, our initial question will be, which species will we obtain our samples? And typically that will be with a mouse. You can use a monkey if you have access to a primate center. And here's a mouse slice, here's a monkey slice. You can obtain them if you have access to a hospital that will provide you with lung tissue from a deceased patient. You can acquire human slices. You'll use a coring tool here that's provided by Precisionary to core the tissue. And there's also other species that have fairly reasonable similarity to the human respiratory system, and that will be a pig and a guinea pig. And most other species would not be suitable for medical research. And there's other rodents, mouse and hamster are good experimental models, but typically the most common and convenient is the mouse. So initially, when we start our experiment, we will have to boil agarose, preferably a low melting point agarose. Precisionary provides that in their package when you buy a compressed dome. But you can also purchase from Invitrogen or Thermo Fisher low melting point agarose, and you will have to boil it thoroughly at a percentage of about 1.6% to properly inflate a lung. And the microwave oven that I'm showing here, I found to be the best, and I strongly recommend. It's only $250. It's a great laboratory microwave oven. It's large, that if you're doing large samples, you could put a whole liter bottle inside. And what it does is it delivers constant heating at low power. So most microwave ovens, they cycle between, if you set it at power 10, it will give you full power throughout the whole time period. If you set it at power 5, it won't give you 50% power for the entire power period. What it will do is it will cycle between 10 and 0. So half the time it will be delivering power, and half the time it will not. It'll just be oscillating between 10 and 0, because so there's no consistent heating. And what happens is the auger blows up in the microwave. It boils over the sides of your container, and becomes very difficult to manage. So you don't want agarose mess in your microwave oven. I recommend this microwave oven because it will deliver, if you set it at level 2, it will deliver 20% power for the entire time consistently. It's used by chefs that want to cook and are serious about cooking, professionally. And this is the only one I've seen on the market that does this Panasonic cyclonic wave inverter microwave oven that I strongly recommend for boiling agarose. So you will boil your agar, and then once you boil, let's say, for a mouse, you did 30 mils of agar with 1.6% agarose in the agar. So you're boiling the agar, and you'd have to boil multiple times to make sure that the all the particulates inside are completely dissolved. If you are not thorough about the dissolution of the particulates in the auger, small pieces will clog up the airways, and you will have a difficult time inflating your lung. So it's very important. So usually, when I boil, I bring the agarose to a vigorous for uh, a minute, or for seconds, and then let it cool a little bit, and let the bubbles die down, and then boil it again, and then let the bubbles die down and boil it again, and do that about 5 times to make sure your agarose is completely dissolved. And then you'll have a nice homogeneous agar to deliver into your inflation of the lung. So once you've made the auger and you've boiled it thoroughly, you take a look at it, put it up to the light, make sure there is no particulates left, and then immerse it in 42 degrees water bath. So just put it in a water bath and give it some time for the auger to equilibrate to 42 degrees. And this temperature is ideal to keep the auger in a liquefied and for inflation, but will not do damage to the lung of the mouse. So you don't want to damage the epithelium, and you don't want the temperature above 42 degrees, because that could do temperature damage to the tissue. So we will then take our mouse. If we have our mouse, we brought it up in a cage, and what we will do is, here I have my notes about boiling the auger. I've already spoken about that. We will put our mouse here in this bell jar, and it's very simple. Just take a 1,000 ml beaker and find a lid, and create a nice platform for the mouse, and you just drop your mouse inside. Or first you immerse your cotton swabs, 2 of them, with isoflurane, and that will vaporize and euthanize the mouse. So drop the mouse in, so that it's on a platform and its feet don't touch the isoflurane cotton balls, and allow the mouse over 5 minutes to it'll be struggling for a few minutes, and eventually it will become immobile, and you will have after 5 minutes you will have a mouse that is recently euthanized. So it's a dead mouse, and then you'll pull it out of the bell jar and spray it with 70% ethanol to wet the fur. Because when you do the dissection, if you've done mouse dissections, you don't want to have fur getting into your when you open the animal. You don't want it getting into the tissue of the mouse. So you will cut your mouse open and expose all of the organs, and cut open the rib cage, and you can see here is what the lung will look like. It's very flaccid and flimsy. It's not inflated, and you will cut away as much of this rib cage as you can, being very careful with your scissors not to puncture the lung. You'll have a beating heart, so you'll have to remove the heart, and then you will cut away this extraneous tissue and fascia above the trachea to completely expose the trachea. So the trachea will have to be cleaned of fascia and be exposed. And you have the other organs here. You want to make sure you remove as much of the rib cage as possible, and open up the area of the upper upper body, so that you can inflate the lung and not have it get punctured by rib cage or interfere with other organs. but yeah, you'll remove the heart and and have some sterile gauze to uh, absorb the blood. So it'll be quite messy, but you try to absorb as much blood as possible, keep it as clean as possible, and then inflate your lung. Now, what I have here is a liver slice. So if you wanted to take slices of another organ, I just want to point out that that's also possible, and just show the contrast that a liver can be cut without any inflation because of the consistency of the tissue. So if the liver is because of its durability and its consistency, it will not need to be inflated with any auger. You can cut it. You can see the hepatic lobules here, and they're very beautiful. And so you can obtain slices from other organs if that's of your interest. And it's just this is just to underscore the point that the lung has to be inflated with auger because of its consistency, which is very flimsy and flaccid. So we need a nice firm lung in order to do the slicing. So what we do is we will take a catheter. We get these from Becton Dickinson, and I thought I had. Oh, here we are. This is an example of the catheter. I removed this component here. It has a needle inside. So you pull this. It's attached and connected into this catheter. You remove all these components, and pull out the needle. So you just have this part, and then attach the syringes, which you'll pour the auger into these syringes to inject into the mouse. And the syringes have to be pulled out, and then the auger will be added in as they are vertical, and the auger will remain liquefied for several minutes because it is at 42 degrees. So it'll take some time to cool, and that will happen as when it gets injected into the mouse lung. But I usually use 2 syringes, 3 ml syringes for a mouse, and fill them completely in the event I have some leakage. So when I start injecting the auger into the animal, you may not have a perfect seal. You may have some auger coming out the back. It's good to have some backup agarose liquefied, ready to pump in if you need extra. 3 mils should be enough, but if you have some backup extra, that's good. And it takes a little practice to enable yourself to manipulate this syringe with 2 hands while you're holding the catheter end and putting it inside the trachea. So what will happen? As the trachea is opened and visible, you will cut a slit in the trachea that opens up about 40%, creates a 40% opening inside the trachea, and insert this catheter tube inside, and then clamp it with an instrument, a stainless steel instrument like this. Once you have inflated the lung, so you will inject the auger into the trachea, and it will inflate the lung. As you can see, this is nice inflation, and you want to see this type of expansion of the lung where the tips come out and they become very visible and prominent. You see nice tips here, and that means you've filled the entire lung with auger and filled it thoroughly. Yeah, so this is you see you see some more lung on the other side, the left lung, but the auger has been injected, and it is inflating the lung thoroughly, nice and thoroughly, nice firm inflation. This is what you want to see. Nice healthy lung, firm inflation in this way. So this is a very nice slide to illustrate what the lung should look like when it's fully inflated. And then you clamp it off, and then pour cold Hank's balance salt solution on the top to wash the lung, and possibly put some ice on top. Please give me a moment here. I need to. Okay, so this is a good example here of a inflated lung. I will go to the next slide. And here's what you're filling. You're filling all this airway here, and the alveoli. And if your agarose is clean and even and very homogeneous and well dissolved, you won't have any clogging. And the agarose will fill up all these airways and inflate the lung, as we see here. So this is this is nice. I like this slide. Okay, so now we're going to make the slides, the slices, and we will cut the lung tissue out of the animal and put it into a petri dish as shown here. And this will be in Hank's balance salt solution, so it has a chance to wash. And you might want to do a couple media buffer changes to make sure that the solution is nice and clear and clean, and you have a nice inflated lung that has cooled. The agar has solidified inside, and it's a nice firm piece of tissue that is floating in Hank's balance salt, chilled Hank's balance salt solution. So this chilled, and it's nice to put this on top of ice to make sure it stays cold and you keep your lung tissue nice and chilled. So then we will let me go back to the slide here. Now we a couple cutting parameters are interrelated and need to be we should explain. One is the inflation pressure. As we pump in the agar, we want to make sure we have a nice firm inflation, and the consistency of the tissue is of it's plump. It has a very plump feel to it. If you're depending on the animal, it's easy to do with mice. If you're doing it with a monkey, this could be difficult to make the tissue nice and firm, and so you will might have to put some more pressure to get the proper consistency of the tissue. And then the other leg of the triangle is the cutting efficiency. So this will depend on the sharpness of the blades, the oscillation frequency, and the speed the blade moves through the tissue. And we'll discuss that as we take the long piece and put it on a pedestal and insert it into the compressome. So here's our tissue. We will sever this and clean this connective tissue here, and we'll have a piece of lung that is nice and firm and ready to apply to the pedestal. We'll cut the tissue along an edge to make a nice flat edge to apply the mouse slice to the compressome pedestal. It's a Teflon pedestal, and here it is. So we will put a little bit of crazy glue. I've tried to use super glue, and I found out every time I use super glue, my slices are not that healthy. There's some component of the super glue that is toxic to the tissue, so I don't recommend super glue. I recommend crazy glue. So you'd want to take some crazy glue and apply a thin layer here, place the slice on top, give it a few minutes to dry, and you'll have a nice piece of tissue that is attached to this cylinder, this Teflon cylinder, that will be used to insert into the compressome. So we will then slide the stainless steel sleeve over the Teflon cylinder, and to an extent that it covers the top of the tissue, and then take some of our agar that is still in the water bath. We will have some left over that we didn't use to inflate the lung, and cover the tissue with it, and allow that to this will allow this to solidify. So it's a nice core of agar with embedded lung tissue, with an embedded lung tissue piece that is ready to insert into the compressome, and this whole piece will be inserted into the compressome. So you can use, if you want a little more firmness to the external agar, you can have a separate agarose that's a little slightly higher concentration. So some people will prefer the internal, the inflation agarose at 1.65%, and then that you might want a external agarose at maybe 1.8%, so you have a firm, but I don't think that's really necessary. Usually 1.65, the same agar concentration that you use to inflate the tissue, is sufficient to put on the exterior of the lung slice, of the lung piece that you insert into the pedestal. Okay, so the Precisionary, they provide a cooling tool here to expedite the chilling of the agar and solidification of the agar. So we will put the cooling tool for maybe a minute or so, and expedite the chilling, and we'll have a nice solidified agar inside the cylinder with the tissue inside that has been inflated, and it will be ready for cutting. And we will insert the cylinder into the compressome on the side, and we will allow the cylinder to fully enter and penetrate the into the water bath, into the collection tray. So here's our, so we've inserted our cylinder into the compressome unit, and extended the this rotating piece to the edge of the pedestal of the cylinder, so that as it rotates, it will push the tissue into the blade that is cutting and oscillating and producing slices, as you see here. So a key cutting parameter is our slice thickness, and that's for any tissue. And for a mouse, I'm finding that you can go down to 150 micron as the thickness of the tissue, and I tend to find that's a little too thin. And a good thickness is probably about 200 micron for a mouse slice. So you would set that on the compressome unit, set the cutting thickness to about 200 micron. You can go up to 225, but 200 micron is really a good cutting thickness. If you go too thin, if you go under 150, you could start fraying the tissue. And I can't imagine why somebody would want a really thin slice. Also, your smooth muscle rings will be very weak because they're so thin. So you may not get a robust or consistent contraction of the smooth muscle rings if you apply contractile agonists such as beta methacholine. So now it's important as you increase the thickness above 150, closer to 200 or 225. Some people can go as high as 300 if they want a really thick slice. And what that will do is that will create a lot of that will create an extensive depth of field and numerous focal planes. So that's the problem with going at too high a thickness. If you have too many focal planes, as you adjust the focus on the microscope when you're looking at your slice under microscopy, it's going through various focal planes, and it's hard to tell what the proper diameter of your smooth muscle ring is, because it will vary somewhat uh, along that 200 micron distance, vertical distance. So that's why I think 200 is probably ideal for a mouse, and people can go as 225. I found for monkey that 225 is ideal, and that would probably apply also to human, because monkey and human are fairly similar. But you don't want the slice to be too thick. That will give you too much of a depth of field, and then you have too many focal planes, and it becomes impossible to obtain consistent readings as to the lumen diameter itself, because like I said, there's variation in the lumen diameter across that thickness. So here we are with our, we'll have a setting of, I usually for mouse, I'll use an oscillation speed of about 50% of what the system offers. So I'll set that at number 5. I'll have a cutting speed at 5. And we'll start cutting the slices as the rotary component turns and pushes the auger out with more tissue for each individual slice, for each individual cut or oscillation of the blade. And you can see these mouse slices here are nice crescents that are cut from the tissue, and you'll have many of them that build up, and probably for a mouse lobe, you may have 20 to 25 mouse slices. So if you were to do all 4 lobes for a single mouse, you may generate 80 to 90 to 100 slices, and that's a good production to have that many if you use all 4 lobes of a mouse. So we will take the mouse. We will take a what I used is a bacterial loop, sterile plastic bacterial loop to fish these slices out of this Hanks balanced salt solution water bath and transfer them to a petri dish. And I'll transfer them to a 100 millimeter dish with about 25 mils of Hanks balanced salt solution, and I'll transfer all the slices. And then you can take a slice out and put it into a smaller petri dish, or into a 6-well plate or a 24-well plate, 12-well plate, whichever you choose, and look at it under the microscope, and take a look at your slice, look at the quality of the slice and how clean it is, and if you have nice smooth muscle rings and they look healthy. But remember that the slice has undergone quite a bit of trauma during the slicing process. And we will transfer all these washed slices that are in Hanks balanced salt solution to a slice media, so a cell culture media that I'll discuss in a minute. But the slices will need about a day, probably 24 hours at least, to recover, or maybe 48 hours to recover from the trauma of the slicing process. So you wouldn't use these for experiments on your first, just after slicing. You'd want to give them some time to recover and rebuild damaged tissue. And I like to go even 4 or 5 days. Give them 3 to 5 days to recover and be a healthy lung slice. And this is how it will look under microscopy. You've seen this slide. So you'll have here lung parenchyma, and you'll have a smooth muscle ring. You might have a blood vessel. Now, what this slide shows is I had some bubbles in my injection. So this is good for illustration. This was one of my first slices that I did. And if you don't have in your syringes when you're doing the injection, if you haven't completely purged the air out of the tubing, you might get some bubbles, and these are unwanted. So these were bubbles that got injected into the mouse lung during the injection process, and they're kind of you know an unwanted part of the microscopy. So it's my first one, and during your first attempt to inflate a lung, you might get some bubbles. And it takes practice, just handling the catheter and syringes and the whole apparatus. So the slice for media that you'll transfer the slices to from the Hanks balanced salt solution is a serum-free media. You'll have no FBS, and it will be standard cell culture media. So you'll use like a Corning or Gibco, DMEM F12, high clone, whoever you buy from. You might inject 1X non-essential amino acids. I like to you'll put some insulin in to make sure that the cells can uptake sugar. For longer term culture, I put about 2.5 microgram per mil transferrin. This is for iron because cells tend to need iron. Some people might add a steroid, such as hydrocortisone or dexamethasone, to allow the cells, but that might confound any experiments that have any steroid related or lipid related treatments. So you have to be careful if you're using a dexamethasone or hydrocortisone. It's not necessary. And of course, you'll want some antimicrobial additives. So you'll want penstrep, gentamicin, and antifungal such as amphotericin. And this is what can happen despite having a antifungal additive such as amphotericin. So these are monkey slices, and the monkey slices, well, you can see they're circular here, not the nice crescents that you get from mice, because we use the coring tool for this. And they this here, what happened was a fungus grew on top of the slices. So the slices tend to kind of float near the surface of the media. And after this took a week. It took a week to get to this. But then you have a fungal infection growing, and you'll have to generally you'll have to dispose of the plate. It will be almost impossible to remove the fungus and preserve these slices once this plate has become infected. Once you take for experimentation, you will put the slices in under microscopy and apply an agonist to contractile agonist to contract the smooth muscle. And for mouse, that will be beta methacholine, not histamine, because for some reason the mouse smooth muscle is not responsive to histamine. Now, when you get to monkey and human, it's the reverse. So the smooth muscle of the human or the monkey will be responsive to histamine and not beta methacholine. So you have to use a different contractile agonist to cause contraction. And so we will prior to the contraction process, let's say I added 5 micromolar. I had Hanks balanced salt solution, and I had a contractile ring of this dimension, with this amount of opening, And we used image J to quantitate the size, the area, and that's using this tool here. So you'll just go File, open your image of your slice, and then click on this selection tool here, and then click around the interior of the slice, and then click Analyze, click Measure, and it will give you a value of the two-dimensional surface area of the lumen itself. And when you apply let's say in the case of a mouse, you applied beta-methylcholine, say 5 micromolar, this will start to contract, and this lumen will start to shrink, and that will happen over minutes. So it might take 5 minutes. You can, as you do a time course, the contraction will increase over time, and the lumen will get smaller and smaller. So by 20 minutes, you'll have a very small lumen. That's maybe this size. It might be 50% contraction. A lot of times we get good 20, 30, 40% contraction, sometimes as much as 60% contraction. And that is a good pharmacological experimental treatment to validate the health of the smooth muscle ring. If you have contractile slices, then they are healthy and they are contractile. They are viable. Another way to measure or assess viability is with a fluorescent assay. So there's a live dead assay from Invitrogen that can be purchased through Thermo Fisher that uses calcine or ethidium homodimer. And when you add the calcine or the ethidium homodimer, if the smooth muscle ring is viable, it will fluoresce green under excitation with a GFP filter. So you'll have a nice fluorescent green for a healthy smooth muscle ring. And if it is dead, the red ethidium homodimer will fluoresce. And in this case, it is not fluorescing. You'll have some background fluorescence scattered around the slice, but because it is not fluorescing red, but is fluorescing green, this is a viable slice, and this is a way you can test your the viability of your slices, of the smooth muscle rings in your slices, to make sure they're healthy. Because sometimes you may, for whatever reason, you may not have contractility, and then you'd want to say, well, are my slices even viable? Are they dead? And you'd conduct an assay like this if you didn't have contractility, just to see if your slices are still viable. If they are viable but they're non-contractile, then it could be your beta-methylcholine was improperly prepared. It's old, or the agonist is there's a problem with the chemical that's being used to stimulate contraction. Or there's something wrong with the slice itself. It's paralyzed in some way. But that will involve some troubleshooting if you have a viable slice that is uncontractile. Now, if this ring here is fluorescent red, it will indicate to you that it's dead. So if your slices were non-contractile and your ring is fluorescing red under a live dead assay, you know why. It's obvious that your smooth muscle rings are dead. And the cells are dead, and that's why they're not contracting. But the nice gold standard is if you have beating cilia. So if you looked under a microscopy and you had nice beating cilia along the interior, then that implies that your epithelium is quite viable. Your smooth muscle ring will therefore should be viable, and you should have contraction. And if you don't have contraction, you'll have to troubleshoot why the slices are not contractile. And if you don't have a complete ring, so in other words, if this is broken here, then the smooth muscle won't be able to contract. So when you select slices for experimentation, you want to make sure that you have a nice complete smooth muscle ring. your chances of having a nice complete smooth muscle ring increase with the thickness of the slice. So if your smooth muscle ring, I mean, if your thickness is 150 micron, you'll tend to have fairly intact smooth muscle rings most of the time. But if you raise the thickness to 200 micron, you'll have intact smooth muscle rings a greater percentage of the time, and that I think that's better. That's why I like the 200 micron thickness, because when as you decrease the thickness of the slice, as I said, you'll increase the frequency of smooth muscle rings that are not intact, that are not 100% have smooth muscle all the way around the perimeter. So you can do cytokine assays. It is also possible to you have your slices in media. You verified that they're viable, they're contractile, they're healthy, and you can do a treatment. Say, give the slice IL-13 or some cytokine, and then look at the the media, and take the media and analyze it under ELISA to see what the smooth muscle tissue is extruding into the media, and there may be downstream. So if you add IL-13, there may be downstream cytokines that are secreted by the tissue, and you can measure those by ELISA. And we've gotten good data by this technique. The You'll have to make sure your slices are roughly about the same size. So just as long as you check your slices and make sure they are roughly about the same size, you should have good. You shouldn't have too much variation in your experimental groups. I just want to share some literature that you can that are nice resources for anyone just getting into slices. You'll want to read these papers. I was trained by these people here, Xinbin Ai and Yan Bai at Harvard University, also Ramaswamy Krishnan. Are all great people, and they've written some excellent review articles on technique and on procedure with protocols. So you can download um, to read. There's other slices. I mean, sorry, other articles here. There's a book, Asthma, that has protocols on precision cut slices in the middle of the book, and we have these Ray Panettieri's group is doing slices. A number of laboratories in the country have required expertise in slices and have published papers. So you can download these articles from the internet and read them. And that concludes my presentation. I hope that gave you some good insight and technical insight into doing this procedure. And you don't have to. Hopefully, taking some of the tips I've given, you won't have to go through all the R&D to figure out how to generate useful slices. So that concludes my presentation. Thank you.
Thank you very much for the presentation. So for anybody who is watching this online, if you do have any questions after viewing this presentation, please email us at info@Precisionary, and we will forward any questions to Ken so he can answer them for you. Thank you again, Ken, and have a great rest of your day.
Okay, thank you. Bye.
Thank you. Bye.